Hello students, welcome to the lecture 3, that is evolution of specialized metabolism from primary metabolism. So, in this lecture the things which I am going to cover about the evolutionary path towards complexity. We will talk about the gene duplication and evolution of specialized metabolites. We will talk about enzyme promiscuity and its role in evolution of specialized metabolism, and also we will talk evolution of new pathways in specialized metabolism. So, these topics are very exciting, and please do not get daunted with these big names and structures. So, you do not have to remember all these things, but at least the what I feel that you should appreciate the concept at this stage. Later we will get chance to study these pathways in details. So, in the last class I ended up with this slide, that is the origin and ecological function of specialized metabolism. So, origin of specialized metabolism is from primary metabolism that we have established. Now, if you see the evolutionary path towards complexity, and what we see here in the left slide, that is a complex network. So, which includes synthesis of nucleotides, amino acids, carbohydrates, lipids, which all of course comes under the primary metabolism, and from there you see the terpenoids evolve, caffeic acid is coming. So, caffeine is alkaloid, caffeic acid is coming on the phenylpropanoids. And if you see the distribution of plants, particularly from the taxonomic distribution, starting from the evolution when plants originate from the water and then it moves towards the land, that is from the aquatic to the terrestrial. So, started with the lower group, that is alga, then bryophytes, then pteridophytes, then gymnosperms, and then flowering plant angiosperm. So, what is showing here that when they started their journey towards the land, at that point of time they started synthesizing some of the specialized metabolites which help them to grow as a sturdy plant. So, that includes the synthesis of phenylpropanoids and some other metabolites like flavonoids. Then we see further when we get more evolved plant groups, for example, the pteridophytes, where you see that typical vascular tissues formation is there. So, that means xylem and phloem, that means the lignin synthesis is there. Then subsequently we will see the true leaf formation, and when there is true leaf formation, a leaf has lot of important function to play, not only to photosynthesis, but also store some of these very specialized metabolites in their epidermal cells, which modified into a specialized structure called trichomes. What we find in case of mint, if you take any peppermint or spearmint, if you touch the leaf, if you rub your fingers and you smell, you will get the typical smell of mint smell in your hand. What is happening there? The epidermal cells of mint contains lot of trichomes. So, trichrome accumulates these terpene compounds, and that is now transferred into your hand, because when you touch you are breaking the trichomes. Similarly, the seed formation, next we see the seed formation in gymnosperms and then angiosperms. So, there the seeds are important, seed has to be stored and it produces some tannins, and then the starch needs to be broken down, and then what we see, the mostly evolved the flowering plant. So, flowers plays important role in pollination, and flower produces lot of metabolites including color and volatiles, and these come under specialized metabolism. And not only that, if you see the legume plants, legume also produces very specialized metabolites, because legumes are the nitrogen fixing plants. Why? Because they contain the nodules in their roots, and this nodulation requires the interaction of the rhizobium bacteria with the plant, and their specialized metabolites, particularly flavonoids and isoflavonoids, plays important role. So, this is how the evolutionary pathway moves towards complexity, and the more complex the plant, more diversification of the metabolites. Now, what we see here, this looks a bit complex at this point to you, but you try to appreciate it. What is this? The first one is talk about in the divergent evolution. So, this leads to the synthesis of flavonoids. So, flavonoids are a group of compounds that produces in that universally produces in plants. A specialized group of flavonoids are called anthocyanins, which is responsible for the flower color. There are of course other flavonoids as there. So, what is showing here, that is a O-methyl transferase that undergoes folding, and that leads to different enzymes in two different plants, and which differ in their active site as you see from here, as well as they have distinct substrate specificities. Okay. Whereas, here in the second one, this is about the lignin. So, here basically the active site is similar, and it is the same substrate specificity, and that is particularly showing in the pteridophyte group of plants. One is from the selaginella and this for the Lycopodium. So, that is called the parallel evolution, and whereas the third one, it is a more complex, it produces flavonones, it produces flavones from flavonones. So, here basically the distinct site in the residue and the substrate specificity is same, but distinct inactive site in the residue. Okay. Next we go. So, once we know that, then again this apparently looks complex, but at the end it will be clear. So, what you see here, that there are cousins of primary metabolism, for example, carnitine acyltransferase, sterol demethylase or catechol methyl transferase. These are basically the primary metabolic enzymes. From there, these enzymes undergoes changes in and diversity in its amino acid position in its active site, and that comes under folding, and eventually that leads to the evolution of acyltransferase group of enzymes. What we see here, for example, HCT, it is a term of, it is a type of acyltransferase which converts coumaroyl CoA to coumaroyl cyclimate. When we will study the phenylpropanoids, we will see this in details. Another one is that formation of this putrescine conjugates, polyamine conjugates, where this acyltransferase plays important role. And another important compound is rosmarinic acid synthesis, that is a marker compound of Lamiaceae plants, particularly the Tulsi family. So, there also acyltransferase is involved. So, all these acyltransferase are evolved from the primary carnitine acyltransferase. Similarly, the cytochrome P450 type of enzymes, that also evolved from sterol demethylase, and that plays distinct function in the distinct metabolic pathways. And the third one is the O-methyl transferase, which also plays important role in distinct pathways. For example, the trifederal spermidine, which is TSM. So, this is tricaffeoyl spermidine methyl transferase, whereas in case of lignification, the caffeoyl CoA methyl transferase, which converts caffeoyl to ferulyl and eventually it forms the lignin. So, that all comes from this primary route, which is the primary cousin, which is catechol methyl transferase. So, in other word, what I mean to say, that these all enzymes basically undergone several folding and changes its plasticity, and that leads, because of this extensive expansion that happens during the evolution in the plants, and that is why as plant evolve, more complex metabolism also evolve with them. So, this is what is showing that a primary reaction, so a existing substrate which converted into a product by the catalyst by the enzyme. Now, this enzyme or which is basically a protein, when the gene if undergoes mutation under selection pressure. So, E forms E prime, and that once it forms a new enzyme or new protein, enzyme protein, which now converts the existing substrate to a novel product. So, in other words, the primary metabolism, there must be a gene which produces the protein which and protein function, and that function basically supports the growth and development of the individual. So, here selection of existing characters is important. No unfavorable changes will be tolerated, any unfavorable changes will disappear, and the purpose of this is basically to maintain the essential function. Okay. Now, these plants are survived in an environment which is not always very familiar to them or not always conducive to them. So, in that point of time there will be their plants are under tremendous selection pressure. So, what we see that plants evolve the new pathway, that is happens because of the gene duplication and amplification of the existing primary metabolite gene, that leads to gene duplication from this. The green one is basically that is the genes for the secondary or specialized metabolism that encodes the protein which function, and that function is basically help the plant to interact with the environment, and this is more flexible. So, it can bear changes, modifications, variations, so that it is more plastic in nature, it has more plasticity, so that it survive well in this hostile environment, and ultimate aim is basically to survive in the environment. So, what we see here that the enzyme catalytic promiscuity, means is the enzyme catalysis, a catalytic reaction which is basically not the reaction for which the enzyme evolved. So, enzyme is evolved for producing one particular reaction, say this A to B. So, E1, that is the main reaction, okay. But it may have a feeble reaction which can convert another A to another product. So, that is called promiscuity, which will come in the subsequent slides. So, what we see here that E1 makes A to B, and this is the enzyme which is converting this, and this enzyme may slightly makes it conformational changes, although the percentage is very less, and once it does that, then it can do conversion of C to D, and when it changes its conformation, it forms E1 prime. But even E1 prime is there, it will not sustain for a longer time, it will try to come back in its E1 position. But this again if undergoes certain mutation or under unfavorable environmental conditions. So, there may be gene duplication, and that leads to changes in the E1. Now, you see the E1 can convert into this E prime 1. So, this E prime 1 is now relatively more stable compared to this one E prime. So, this now converts C to D and this is about 7%. And so, this is how this is evolving, and eventually the E2 will come here, and E2 can convert C to D. So, the E2 is basically now is the promiscuity. So, eventually if you see the E1 is basically for converting A to B. Now, from E1 E2 is evolved. So, E2 is basically converting C to D, but it has very feeble or no activity of A to B. This all happens because of this gene duplication. Now, this is an existing pathway A to E, Q to T and G to L. So, what you see here the E2 in this pathway it undergoes promiscuity and form the E2 prime. Similarly, E7 forms E7 prime and E11 forms E11 prime. These all three are promiscuous enzyme, and once this formed, it may get a suitable substrate which converts W to X, then X will be accepted by the new promiscuous enzyme which converts X to Y, and then eventually Y to Z by the third new promiscuous enzyme. So, like this way a new pathway entirely can be created from the pre-existing pathway, and that happens because of the evolution of this promiscuity of the existing metabolic pathway enzymes. Let us see the example of DHS. DHS is deoxyribonucleic synthase, which plays important role in basically the activating of the initiation factor required for translation. So, this is required the lysine, and it accepts the lysine, and with the help of spermidine it undergoes activation, and that subsequently help to proceed the translation. So, the gene is called DHS, deoxyribonucleic synthase, and these over the evolution this undergoes gene duplication, and what we see HSS homospermidine synthesis is formed. So, HSS is formed in a particular group of plants. Those plants need to survive in the environment, particularly from the severe herbivore attacks. So, they need to synthesize pyrrolizidine group of alkaloids. So, one example is senecio. I mentioned about senecio, the ragweed, the senecio species. There you will find this pyrrolizidine alkaloid senecio, and that is formed from the homospermidine. So, homospermidine is the precursor of the senecio alkaloids or the pyrrolizidine alkaloids. An example is phenecio. So, homospermidine is basically formed from putrescine, with the help of spermidine by the enzyme which catalyzes the reaction, which is called homospermidine synthase. So, Thomas Hartmann and his group basically they first elucidated this homospermidine synthase, and they characterized the enzyme. So, and they characterize the whole pathway, and this is very important because this is the starting point, and then they try to find out the what basically makes homospermidine synthase comes into the picture. So, they did lot of molecular analysis including site directed mutagenesis, and eventually their results they concluded that homospermidine synthase is basically evolved from the DHS, deoxyribonucleic synthase, by gene duplication, and once this is formed, this no more accepts the original substrate, which is basically the initiation factor. It is not binding, this is lysine of it, instead it performs this reaction. So, this is the existing pathway, and this is what is evolved. C is the new enzyme, and it makes the new product, which is B prime. So, B prime subsequently will make the pathway pushed, eventually leads the compound D. So, here C and D both will be the promiscuous enzyme, and C is basically C's main function is D to D. So, C cannot make A to B anymore. C always has to catalyze the reaction A towards B. So, this is the primary metabolic enzyme. A to B is the main activity, and the promiscuous activity is A to B prime. That over evolution what we see, A becomes A is the substrate for B prime. So, this is what it is, the reaction is telling that. So, this is the pathway for producing the eukaryotic translation factor. Now, over evolution it produces homospermidine synthase, it is homospermine, and that actually leads to the formation of pyrrolizidine group of alkaloids. So, this is the senecio plant, and why it happens? Because it happens because to survive against herbivory, and interesting point is this, that this senecio is also being taken up by a specialized group of insects, and they consume this and they accumulate this alkaloid, and as a result of that, that specialized insects can protect themselves from other big predators which can eat them. And this homospermidine synthase is basically it is produced in the root cortex and the pericycle, and then the pathway starting there, and then the pathway moves towards the aerial part. As I said that long distance transport, this is also a good example of that. And this is the specialist insects which is usually feeding on that ragweed. This is called cinnabar moth, and it accumulates the alkaloid and it is protecting itself from the predator. Next, come to another example. Here a primary metabolic ancestor, this is the enzyme which accepts one substrate and produce a specific product. Now, the over evolution, the gene duplication followed by divergence occur. Now, these primary metabolic ancestor enzymes becomes or it evolved into a specialized metabolic enzymes which is having multifunctional enzyme activity. And what we see here, that cycloartidyl synthase, which is basically the pathway of sterol biosynthesis, that eventually this enzyme can accept the single substrate and makes 3 different products, all related to triterpene biosynthesis. Now, this evolved in different plants, but the basic enzyme is which is the this synthase which evolved from the primary metabolic enzyme. So, over evolution it accepts the same substrate, but depends on the plants, it produces a range of diverse products. Now, let us come into the story of the tropane alkaloids. So, the tropane alkaloids basically synthesized from the putrescine. So, normally putrescine is a polyamine, putrescine is converted into spermidine by the spermidine synthase. Spermidine synthase, and spermidine is utilized in the primary metabolism. So, it acts as a sometimes like a plant growth regulator. So, this is very much a primary function. Now, in the tropane alkaloid pathway, what happens that putrescine, the enzyme which is putrescine N-methyl transferase, which evolved, and that is a specialized metabolic enzyme which takes putrescine as the substrate and makes N-methyl putrescine. So, in here there is the methyl donor is the S-adenosyl methionine here in case of putrescine methyl transferase, whereas the original enzyme which is spermidine synthase, which uses decarboxylated SAM. Okay. Now, and this is the first enzyme of the pathway, and eventually it moves to the tropane alkaloids. So, when you look the tropane alkaloid pathway and genetic manipulation, we will study this more. But interesting point is this, a single amino acid substitution basically converts spermidine synthase to putrescine N-methyl transferase. So, that means over evolution just a single amino acid change makes entirely a new specialized enzyme for making a new pathway to cater specific function. This is another reaction. So, for example, the acridone alkaloids. So, acridone alkaloids are formed from chorismate through anthranilate, and anthranilate one route goes to acridine alkaloid synthesis, another route goes to the tryptophan. Now, the enzyme anthranilate synthase having 2 units, anthranilate synthase alpha 1 and alpha 2. So, the alpha 2 is basically plays important role in controlling, so that the majority of the anthranilate synthase utilizes chorismate, makes anthranilate, and then that moves towards tryptophan, and that is basically and once excess tryptophan is produced, that is being sending the signal, and that is not allowing the anthranilate synthase. Now, this particular control, allosteric control is basically removed, or this deletion, because how it is removed, because there will be deletion of this a part of their active enzyme, and that actually removes the allosteric control, and then what happens, earlier what tryptophan used to control this. So, that control is no more there. So, what you see that the substrate produces the intermediate anthranilate, then anthranilate can goes for acridine alkaloid production, that anthranilate can further converted into production of tryptophan. So, this is how again the reaction of primary metabolism, it is basically changes and leads to formation of the specialized products. Now, let us see one example, like a plants survive in this hostile environment. Directly they can synthesize chemicals which the insect will come, eat them and die. The other way is that when insects are attacking the plant and the plant is sending the signal which is activating a second group of insects which will come and kill the first group of insects. So, this is called tritrophic interactions. So, in this case what happens, when the first group of insects attack the plant, it basically starts chewing the leaf. As a result of that, there will be activation of a pathway leading to the formation of indole. Indole is a volatile compound. So, that basically gives signal to give the to call the another group of insects to come. Whereas, but then this indole 3 glycerol phosphate, that is basically the substrate which is there for making the tryptophan. So, from there will be 2 gene duplication, one leads to indole and another leads to benzoxygenone pathway. So, this one which is the benzoxygenone pathway, that involves the several enzyme groups, whereas the indole it is only the one. So, if we see the next one, so next one here you see that this one is the first gene duplication which leads to the formation of the indole, emission of the indole, whereas the another gene duplication which leads to the formation of benzoxygenone pathways. So, it is clear from this one, the first one producing indole and and this indole in some plants it emits, but other group of plants it is basically converted further into DIMBOA and DIMBOA, which has benzoxygenone and which plays important role in the accumulation, and that helps to defend the plants against other predators and pathogens. But these indoles, this same substrate can also be used for tryptophan biosynthesis. So, this is what it is showing here, that is the maize plant which is being attacked by beet armyworm, and it emits the, once it chews it emits the volatile indole, that is basically calling the other group of insects which will come and lay eggs over it, and basically eventually it will make them dead. Okay. With this, now let us move into the another group of compounds which is called benzophenones, which is very specialized compounds, and benzophenones are basically formed in 2 families, Clusiaceae as well as Hyperiaceae. So, these are the good precursor for different drug compounds. Now, scientists they have evolved they have basically worked out the xanthone biosynthesis. Benzophenone is the precursor of xanthones. Now, the enzyme is basically type 3 polyketide synthase, which basically condenses with either malonyl CoA and benzoyl CoA and makes either benzophenone or the other one which is biphenyl, whereas the malonyl CoA can joins with coumaryl CoA and makes either chalcone or stilbene, depends on the plant. Okay. And the biphenyl basically serve as a precursor for aucupenin and subsequent other products, which basically accumulates in the rosaceous plants as a defense compound, so as to protect themselves from herbivore and other attack. One interesting thing has happened. So, the enzyme which is called benzophenone synthase, which which joins malonyl CoA and benzoyl CoA and makes basically the benzophenone, which is the starter for xanthones. Scientists characterized this gene and they wanted to study more about it. They wanted to see that how basically the benzophenone evolved. So, the strategy they have used, they use site directed mutagenesis, and while doing a range of experiments, finally they were lucky enough to find one side directed mutant, which they call this threonine 135 leucine. That is 135 position threonine was replaced by the leucine, and that eventually leads to a new enzyme activity, which makes phenylpyrrone instead of benzophenone. So, another interesting point is this: in order to make benzophenone, it requires 3 malonyl CoA, whereas phenylpyrrone it requires only 2 malonyl CoA. Now, if you see this, what happens? This is the benzophenone synthase. It makes benzophenone synthase enzyme, which accepts the substrate and makes trihydroxybenzophenone, but it has a very feeble activity of phenylpyrrone formation. Now, when the recombinant enzyme discovered phenylpyrrone synthase, that basically now accepts malonyl CoA and 2 molecule benzoyl CoA, and it makes phenylpyrrone. And when they characterize this enzyme, they find in the literature that phenylpyrrone products are known to form in the primitive vascular plants, which is called Xylotum nudum. It is a very primitive pteridophytes. So, there xylotinin, a compound which is basically phenylpyrrone derivatives, it accumulates there. Another interesting point is that this not only accepts benzyl CoA, but it can also accept the acetyl CoA and make subsequent substituted products. Now, interesting point is this: the proposition now put forward that from primitive vascular plants the complex vascular plants evolved. So, primitive vascular plants making the xylotinin. So, likely that phenylpyrrone synthase or a related enzyme is basically there in the primitive vascular plants, from where with the complexity the benzophenone synthase evolved for performing specific function. This is really very exciting, and this was basically the outcome of a project called evolution of metabolic diversity. So, such program ran for 6 years in Germany, and this is the group which basically discovered this work. Okay. And now we come to the another aspect, which is evolution of new enzyme in the specialized metabolic pathway. Now, what we have said in the earlier slides, that a primary metabolic enzyme undergone gene duplication and form the specialized metabolic enzyme. So, here the concept is different. It's just talking about the new pathway. So, there is an existing pathway A, B, C, D, E, and there is, and what has been known that V, W, X, Y, Z. But what scientists discovered is a new enzyme which connects C to X, and subsequently after this discovery, what scientists have established that indeed the Z is formed not from V, but from A, B, C, X, Y, Z. So, this is basically the new pathway which is elucidated upon the discovery of the new enzyme here. So, this is in connection with the coniferyl aldehyde 5 hydroxylation, which I will show in the next slides, and discovered in 1999. Another one, say there is an existing pathway say V, W, D, E, okay. And there is another pathway exists which makes A to B, B to C, but after C it moves either D or to some other direction. A new enzyme discovered. So, it is not rather discovered, basically it is a heterologous enzyme. When it expressed in a plant system, what it happens, it basically utilize the substrate C and channelize the product instead of D towards Y and Z. So, this new enzyme is basically creating a new pathway for formation of Z. This is called benzenoid pathway, and this was published in this journal, So, this is what it is, the monolignol biosynthesis. So, the upon the discovery of this new enzyme, which is CALD5H. So, this converts coniferyl ferulic acid, ferulic acid converted into coniferaldehyde, ferulic acid converted into converted into 3,4 dihydroxy coniferaldehyde, and then subsequently by a methyl transferase it converted into synapyl synapyaldehyde, and then these by the action of CAD it makes synapyl alcohol, and the existing pathway is this. This makes ferroalcohol, it makes coniferaldehyde it makes coniferyl alcohol. So, earlier proposition was this, that this formed from this way, but the new proposition said no, this is formed from this way. So, what I said, A to B, B to C, X to Y, Y to Z. So, this is the one, and the other one what I said, that upon the expression of the heterologous gene, which is HCHL, which basically channelize the pathway in a new direction. Normally, this coumaryl CoA is the substrate which makes flavonoid, chalcones, monolignol. Upon HCHL expression, this coumaryl CoA is diverted to a new route leading to the formation of benzenoid compounds. So, this is also an example of creation of new pathway of benzenoid biosynthesis. So, to conclude, there are plant produced hundreds of thousands of moles molecules which are called specialized metabolites, and this happened in plants as a consequence of diversity and rapid evolution of specialized metabolic pathways. And these novel biosynthetic pathways basically originate via duplication, gene duplication, or by functional divergence of the existing genes. So, with this basically I will end this class. So, where what we have seen, that the specialized metabolic pathways originate from primary metabolic pathways. So, the genes of primary metabolic pathways evolved, undergoes gene duplication and subsequent divergence, leading to the formation of new specialized metabolic genes. Eventually, it codes the enzymes, and that catalyze the specific reactions for specialized metabolism. So, in other words, the specialized metabolism is not a discrete event, it is basically the extension of the primary metabolism only. So, with this I am ending this class, and in the next class we will talk now more about the biotechnology aspects. So, these specialized metabolites of course useful to us, as I said, for various pharmaceutical and other cosmetic applications. So, we will look into this, we will see how biotechnological interventions in plants can lead to the production of these metabolites, and what are the ways these metabolites can be analyzed. So, that we will see in the subsequent classes, and how to enhance the metabolites levels by doing empirical manipulation, that we will see. And once we finish that, then we will look into the more fundamental aspects about the metabolic pathway, how can we manipulate the pathway, how which will be the metabolic engineering approaches. Okay. Thank you very much.